This is the second of the Unit 6 Biology revision videos, and this will be looking at fungi and viruses. Fungi are another large kingdom of organisms. They cover organisms such as mushrooms, microscopic fungi, yeasts and moulds. Now, yeasts are really important to us because we use them commercially in the making of bread and also beer. So we're going to focus on yeast. Now, yeast cells are a lot larger than bacterial cells. They can be easily seen under a light microscope, whereas bacterial cells appear very, very small even at the highest magnifications. If you wanted to see the internal structures of a yeast cell, you would have to use an electron microscope. This is because the internal structures are really quite small, and we would need that extra magnification that allows from the electron microscope to be able to view those. Now, fungal cells are quite strange actually, because they've got a lot of things in common with plant cells and again a lot of things in common with animal cells. It has a cell membrane, a nucleus, cytoplasm and mitochondria, which are all found in both animal and plant cells. However, it's got a vacuole which is only found in plant cells, but also obviously in fungi such as yeast. And here we've got cell wall, which in plants is made from cellulose. In bacteria, as we said in the last session, is made from peptidoglycan. But in fungi, such as yeast, it's made of a substance called chitin, which you may have heard of before when we've looked at exoskeletons of insects. This cell wall provides the same function as it does in bacteria and plants. As I say, the only difference is the substance that it's made out of. If you cast your mind back to when we did the revision session on bacteria, you'll remember that bacteria undergo reproduction via binary fission. Now, yeast undergo reproduction slightly differently. They do it by a process called budding. This is asexual reproduction in the way that no DNA has been swapped between cells. As in any cell replication, the nucleus and the genetic material has to duplicate first of all. After the DNA has been replicated, the cell undergoes budding. Now, this is an example of a cell in this picture where budding has started to happen. We can see this here by this part of the cell which has started to grow outwards from the main body of the cell. Now, you can see in this cell here that budding has become more advanced, and this cell is becoming more the size of this one. In the end, these two cells will become two separate cells that are genetically identical, but they will still be attached like we've got here. As in bacteria, this way of reproducing, this budding, like bacterial binary fission, it's a very fast, efficient way of replicating, so we can get many cells very quickly. Now, we've got different factors that can affect the growth rate of yeast and this budding process. These factors are the availability of food for the cell, the temperature, the pH, and the amount of waste products that are surrounding the cell. Having a good availability of food will allow for molecules to be built within the cell when it's dividing, things like proteins and the chitin. Temperature, if it's high enough, it will allow for a faster rate of reaction, up to the optimum temperature obviously, where if it goes much over that, we will get denaturation of enzymes. When it comes to yeast, every 10-degree rise in temperature allows for a doubling of the amount of yeast cells. Now, as we said, they do have an optimum temperature, and that is only true up until that point, and then the enzyme will denature. In a similar fashion to the temperature, we've got pH, which if it's too acidic or too alkali, we will get denaturation of enzymes. And the amount of waste products, if there is a lot of waste products building around the yeast cell, these waste products are poisonous to the yeast, so it will kill off the yeast cells. So if this is too high, then we will get less yeast cells in the culture. The last group of microorganisms that we are going to look at in detail are the viruses. Now, these are so small that they were never seen until the development of the electron microscope. They are a lot smaller than bacteria or fungi. In fact, they're about 1/100th the size of a bacterium, which, as you can imagine, is extremely small. Now, if you look at this diagram of a virus, you'll notice that it doesn't have the cell structure that a fungal cell or a bacterial cell has. It lacks a lot of the basic structure that some biologists believe would make it living. So some biologists would class it as a non-living object. In fact, viruses are so simple that really the only things that they're made up of is this coat here around the edge, which is a protein coat, and inside we contain the genetic material, which can be either DNA or RNA, which is slightly different to DNA. Now, because they don't have the cellular structure that bacterial and fungal cells have, they can't actually duplicate their own DNA, which means that they cannot reproduce by themselves. They can only live inside another organism. Now, because it's living inside another organism, we will call that organism the host. Now, viruses are so specific that they will only infect one type of host. Now, this is not strictly true. There are exceptions, such as swine flu and bird flu, where we've got different hosts, but they are the exceptions. But in general, this doesn't tend to happen. So if you've got a human virus that infects a human host, it cannot invade a plant cell, for example. However, animals, plants, and even bacterial cells have all got viruses that will infect them. Once inside the host cell, whether it be a plant cell, an animal cell, or a bacterial cell, the virus will take over, and we'll look at how that happens next. Now here we've got a virus that is on its host cell. Now it's attached to the cell and it's ready to undergo its invasion. Now what happens is, when it's ready, the virus will inject its DNA into the cell. Now remember that this host cell will be extremely specific to this virus. So if this is a virus that infects a plant cell, then this would be a certain type of plant that it would infect, something like a rice plant. Now the genetic information that the virus has put into the cell will replicate, and the genes will cause the host cell to produce the components of the viral structure. So as you can see here, we've clearly got the protein coat of the virus, and these green strands here are the genetic information that has been injected into the cell from the virus. Now, as you can see here, in the last stage of viral replication, the host cell has actually assembled the protein coat and the genetic information into the viral molecule. Now, what happens is these build up in such large numbers within the cell that the cell will burst and release all of the what we call progeny viruses out of the cell. Now, this could be hundreds or thousands of viral particles, and they will all exit the cell and go on to infect nearby cells. And then the same process would happen again, and eventually you get the death of lots of host cells, and hence the onset of infection.